Bismillahirrahmanirrahim. 9700 A-level biology, June 23, paper 1-3, question 1 to question 20. As we all know, this paper now is for 1 hour 15 minutes, so the difficulty level has greatly increased. So one must be very aware of that. Since June 22, the syllabus changed. So this is going to be a new thing that this MCQs are going to be more difficult. Question number 1, which feature is visible with a light microscope using a natural light source? Now you see the wavelength of light is 400 nanometers. So peak to peak, this is 400 nanometer. Now anything less than half the wavelength, so anything less than 200 nanometer cannot be seen with the light microscope. So this is nanometer, this cannot be seen, this cannot be seen, this cannot be seen. So all these answers are wrong. The only thing seen is B, which is something which is a diameter of 200 micrometer. So 200 micrometer means it is bigger than 2, because 1 micrometer is equal to 1000 nanometers. So it is 200,000 nanometers. So anything less than 2, well, the reason why you can't see anything smaller than 200 nanometers is because it doesn't interfere with the light waves. So anything more than 200 nanometers will interfere with the light waves, and that will be possible to be seen. Then coming to question number 2, the electron micrograph shows a structure found in the cytoplasm of an animal cell. Which biological molecules are found in this structure? You all know this structure is the centriole, and the centrioles are made up of microtubules. So basically, they're made up of proteins. So the answer 2 and the answer being D is only proteins. Then which cell structure contains nucleic acids? Cytoplasm, lysosome, and mitochondria. Well, cytoplasm will contain nucleic acid. You see, when they're asking nucleic acid, you have to think is DNA, or you can think of RNA. Now, in the cytoplasm, if you remember, there are free ribosomes, and the ribosomes are made up of ribosomal RNA and protein. So, in the cytoplasm, there will be the free ribosomes. So, nucleic acid will be found in the cytoplasm. In the lysosome, it contains hydrolytic enzymes. It is a phospholipid bilayer. It's a single membrane bound organelle. So, not in lysosome. Mitochondria, yes, has circular DNA, plus it has 70S ribosomes. So it is the answer to the question number 3 is C, 1 and 3 only. Then it says mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. So mitochondria, so a prokaryote means a bacteria ingested by and then have been evolved from prokaryotes that were ingested by an ancestral cell. Which features have prokaryotes lost during their evolution into mitochondria? You see, we have to understand is which features what are the present features of prokaryotes? They have a cell wall, but mitochondria does not have a cell wall. So that is why the answer is A, that they have lost the cell wall. But why? As you see, circular chromosomes were present in bacteria, but they're also present in mitochondria. Now, endoplasmic reticulum was never present in bacteria. So it is not something which was present in bacteria and then they have lost it. Endoplasmic reticulum, it says which features have prokaryotes lost? Prokaryotes do not have any endoplasmic reticulum. Yes, they have ribosomes, and those ribosomes are still present in the mitochondria. So that is why all these answers are wrong. Everybody must be very clear why these answers are wrong. Then coming to question number 5, which polymers are present in all viruses, all prokaryotes, and all eukaryotes? So that means viruses means all the ones which they have a nucleic acid, DNA or RNA, they have a protein coat, some have a phospholipid bilayer outside it. Prokaryotes all have a cell wall made of peptidoglycan, they have a cell membrane, they have cytoplasm, they have 70S ribosomes, they have circular DNA. All eukaryotes means all plant cells, all animal cells, all fungus cells. So which polymers are present in all viruses? Now question number 5. The question, it is polynucleotides, that is they can be DNA or RNA, and polypeptides, they can be protein. You see, viruses is the one which is the confusing one. All viruses will have a protein coat and a DNA or an RNA strand. So polynucleotides and polypeptides was the answer, and that is why the answer is B. Then it says question number 6, how many of the listed structures typically contain genetic material that has telomeres? Bacterial cell has a circular, you see telomeres are only present in when there's a linear DNA, and these are the ends of the chromosome. So it has to be linear DNA, it has to be chromosomes in pairs. So how many of the listed structures typically contain genetic material? So bacteria has a circular DNA, chloroplast has circular DNA, mitochondria has circular DNA. So it's only one part. So that is why the answer is only one of these listed structures. You see, these are 4 listings. 1, 2, 3, 4. How many of these, how many of the listed structures contain genetic material that has telomeres? Some of you don't understand the English. Then it comes to question number 7. Steps 1, 2, 3, and 4 are used to test for a non-reducing sugar. Put 5 centimeters of solution into a test tube, add a few drops of acid, neutralize with alkali, add 66 centimeters of Benedict solution. So naturally you have to heat it after the Benedict solution, so it has to be after step 4. But then you have to heat it also with the HCl. So that is why the answer is B, between steps 2 and 3 and after step 4. You see, most of you would have done D because after step 4 you remembered that Benedict solution is heated. But then you didn't remember that when you add the HCl, so heat to hydrolyze and then cool it and then add the alkali. So maybe if you had forgotten that, then you would get this wrong. Then question number 8, which features contribute to the function of a cellulose molecule? Cellulose is a cell wall. It's very, very rigid. It prevents the cell from bursting. So long chains of glucose molecules into a helix. Many hydrogen bonds form between adjacent chains. It is insoluble in water. You see, long chains of glucose molecules into a helix. No, there is no helix in the cellulose. So what you have to understand is that this would be a little bit of a distractor and you won't really know whether you knew this or not. So question number 8, the answer is C, 2 and 3 only. Many hydrogen bonds between adjacent chains and the fact that is insoluble. So it does not affect that means the cellulose cell wall can function as a rigid structure, preventing the cell from bursting in a plant cell. What correctly describes triglycerides? Non-polar or polar? First, look at the first sentence. You know, fats cannot dissolve in water. They are insoluble in water. So the fact that if they were polar, they would have dissolved in water. So they're not polar. So C and D are perfectly wrong. So they are non-polar molecules that are soluble in ethanol. So 9, the answer is A. They are not soluble in water. The fact first they wanted you to know whether you knew what were triglycerides, they're fats, the fact that they are non-polar, the fact that they do not dissolve in water, and the fact that they dissolve in the organic solvent ethanol. Then coming on to question number 10. Now this is a little confusing and I would like to explain it a little bit. In this question it says cocoa butter contains 3 different triglycerides. These triglycerides are made from the fatty acids oleic acid O, palmitic acid P, and stearic acid S. The 3 triglycerides found in cocoa butter are POS, SOS, and POP. Now, how do you handle this question? It's a little difficult, the reading part, but otherwise it's pretty easy. The chemical structure of the triglyceride is shown next to a diagrammatic representation of POS. So P has 2 carbon less than O and S, and O has a double bond. So P has 2 carbon less than O and S, and there is a double bond in O. So that's a unsaturated fatty acid. P and S are saturated fatty acids. Now look at the question. Triglyceride POP contains 2 unsaturated fatty acids joined by ester bonds to glycerol. By ester bond to glycerol is fine, but then POP 2 unsaturated. There's only one unsaturated in O. P is saturated. Another P would also be saturated. So that is what you have to understand. Then it said triglyceride POS contains 2 less carbon. You see the 2 less carbon is in P. It contains 2 less carbon than POP. POP will have 4 less carbon and POS will have 2 less carbon. So what we have to understand is Which one is correct? Triglyceride POS contains 2 less carbon than triglyceride POP. POP will have 4 less carbon. Then it says triglyceride SOS contains 4 more carbon atoms. Yes, S has 2. S has 2 more than P. So SOS contains 4 more carbons than POP. So the answer is actually C. Then triglyceride SOS contains 2 unsaturated. You see, SOS contains 2 saturated fatty acids. S is saturated, O is unsaturated, and S is again saturated. So SOS contains 2 saturated fatty acids. That's right. But then this is wrong. Joined by hydrolysis to glycerol. Is joined to glycerol by condensation. A water molecule exits. If you add water, you'll break this bond. Hydrolysis means adding water. Hydrolysis means adding water. Condensation means removing water. So this was wrong. And this is why you have to understand this question was a little, you have to just read it again and again. You see, what you have to understand is that POS has 2 less. There are 2 less here, P में. So POS has 2 less, but if there is POP, then it will be 4 less. So they have compared POS with POP. So POP will have 4 less, POS has 2 less. There are 2 less in P. So if there's another P, then it will be 4 less. There will be 2 less here as well. Question number 11. Which molecule contains at least 4 double bonds? Now, as you know, every amino acid has a double bond, C double bond O. So if hemoglobin and collagen has loads and loads of amino acids, so they will have at least 4 double bonds. Now, on about unsaturated triglyceride, yes, unsaturated triglyceride means 3 fatty acid chains are unsaturated. So there's double bond in that, and there is then one when it attaches to the glycerol. So there will be 4. So the answer to 11 is D. Then question number 12. The diagram shows the amino acid glutamic acid. What is the R group for glutamic acid? So you see, basically NCC, H, and then there's an H here, and then there's a double bond O, and then there's an OH, and whatever is attached to the central carbon, that is the R group. So they said, what is the R group of glutamic acid? Now, very easy. This is this. The central carbon, this is NC, this is the central carbon. So this is the R group. This is the COOH. This is the NH2. So the answer is D. Now, question number 13. The diagram shows three interactions that hold protein molecules in shape. Interaction 1, interaction 2, interaction 3. You know the interaction 2 is a disulfide bond, which is a covalent bond. Then interaction 1 is a hydrogen bond. And interaction 3 is between a positively and negative charge. So it is a ionic bond. So that is why the question number 13, this is in the book. Please look at up this page and then understand this bonding. So the answer is B. Question number 14. Some animals produce antimicrobial proteins which protect them from pathogens. Some animals produce a protein which protects them from invading microorganisms which are called pathogens. These proteins could be used to kill human pathogens. So we can use these proteins and we can give them as medicine to animals to treat these human pathogens. However, when used as a medicine, they are broken down by protein digesting enzymes. So if they're given as a medicine, then you have the famous protein digesting enzyme pepsin in the stomach. But they haven't said anything about the enzyme. They've said it's a protein digesting enzyme. Replacing one of the amino acids found in the protein with an amino acid that has been synthesized in the laboratory resulted in modified protein that was not broken down. So the pepsin was unable to break down this protein. Why? Why was that? Why did pepsin not work on it? So then you have to think, how does pepsin work? It's an the substrate has to have a complementary shape to the active site. So what could explain why this modified protein was not broken down by the protein digesting enzyme? Number one, the modified protein has a different tertiary structure than the original protein, right? The modified protein is not complementary in shape to the enzyme's active site. That's why that enzyme is not working on it. The modified proteins are unable to induce a fit with the protein digesting enzyme. Yes, so this is the protein digesting enzyme which works, right? The new protein doesn't fit this. So what was the substrate like this? The substrate that this was the protein which fitted this pepsin molecule. Now, if this protein is now slightly modified, is like this, then it won't be broken down by this protein digesting enzyme. So that is why the answer to this is 14 is A. All three was correct. Then coming to question number 15, a student investigated the effect of substrate concentration on the rate of an enzyme catalyzed reaction. A graph was plotted to show the relationship between these two variables. The student was asked to take readings from the graph that could be used to determine the Michaelis-Menten constant Km for this enzyme. J, K, L, and M show points read from the graph which the student could use to determine the value of Km. Now you know Km is what? Half Vmax. So half Vmax is L. So this is Vmax. But we had to find out the half Vmax and then you draw a line here. So this is K. So basically you have to do K and L. So that is why the answer is C. So Vmax was J. Half Vmax is L, and then K would be the, how you read this off, you read this and then you read it off with the substrate concentration of substrate here. So K and L was the answer to this. Now coming to question number 16, the end product of a metabolic pathway can act as a competitive inhibitor. This is called end product inhibition and allows a cell to control a metabolic pathway. The diagram shows a metabolic pathway where the end product could act as an inhibitor of enzyme W. Now it says what would be the effect if enzyme Z was inhibited? So we have another diagram. Now we have something like this. Instead of this is wrong. This is correct. Cut it off. Now the end product is inhibiting enzyme Z. What would be the effects if enzyme Z was inhibited by the end product instead of enzyme W? So intermediate 1 has to increase because now this inhibition is lost. So substrate to intermediate 1 will, of course, intermediate 1 will increase. And you have to find out the quantity of intermediate 1 and the quantity of the end product. If Z is inhibited, then end product will be less. So that is why the answer is this. Quantity of intermediate 1 will increase because now that is not being inhibited. Now W is not being inhibited. So the answer is A because the quantity of intermediate 1 will increase. This will increase because now this inhibition is lost. And Z is inhibited, so end product will be less will be formed. Now question number 17, which row shows the distribution of cholesterol and the carbohydrate chains of glycolipids and glycoproteins in a cell membrane? Now as everybody knows, glycolipids and glycoproteins are on the outer surface and the cholesterol is between the phospholipids. So the answer has to be D. There couldn't be any ambiguity about it. Glycolipids and glycoproteins are always on the outside. So why would anybody get this question wrong? I don't understand. I mean, that's what my little understanding is. Then question number 18, which row correctly describes all positive relative concentrations of a substrate of a substance when the substance is moved by endocytosis or exocytosis? Endocytosis concentration is equal, exocytosis concentration equal, concentration equal greater inside or greater outside, concentration equal greater inside or greater outside. You see, this is an active process. It's not a matter of concentration. If something has to be thrown out of the cells, exocytosis, something has to be taken in endocytosis, phagocytosis. So the answer is B, because you have to understand is that this is not something which is down the gradient, like diffusion is down the gradient, osmosis is from a higher water potential to a lower water potential, facilitated diffusion is also higher concentration to lower concentration but through a channel protein. So these are all very active processes. So it could be, it's not a question of down the or again, but basically it's mostly all are against the concentration gradient, but not in endo and exocytosis. That's only in active transport. Then coming on to question number 19, which statement about simple diffusion is correct? It requires specific molecules in the cell membrane. It is a passive mode of transporting substance. It always requires a membrane for transport of substances. It only happens in the cells of prokaryotes and eukaryotes. Now everybody knows that it is a passive mode of transporting substances. Other thing, everything else is, there are no specific molecules in the cell membrane. Requires a membrane. You smell perfume. Diffusion, you can even study diffusion, dropping a drop of ink in a beaker of water. So that is also wrong, that it requires a membrane. Then it only happens in the cells of prokaryotes and eukaryotes. So it happens anywhere. Plant cells with the same water potential in the cytoplasm are put into one of three different concentrations of sugar solutions: 10%, 5%, and 2.5%. The cells were left for 50 minutes and then observed using a light microscope. Cell X, cell Y, and cell Z. You can see cell Y has become more distended and is more turgid. Cell Z has lost the water, it has plasmolized. Cell Y has a lower potential than the sugar solution it was put into. Yes, because water has moved in from a higher water potential to a lower water potential into the cell. The cell has become fully turgid. Cell Z was put into a 10% sugar solution. Yes, because it has moved out, so it must be a very concentrated sugar solution. So water has moved out and it has plasmolized. Cell Z has a less negative water potential. So this is, say, -5 and outside is -10. So that is why water has moved out. -5, -10, less negative to more negative. Less solute means more free water molecules, higher water potential. So always write some figures on it, to do some homework on it to make you understand. So that is why the answer to question number 20 is A, 1, 2, and 3 all were correct. This completes the first video, which is question 1 to 20, and thank you very much for watching and thank you for subscribing.